Good afternoon. Welcome to today's conference call. At this time, all lines are on listen only for today's conference until the question and answer portion of our call, at which time you will be prompted to press star 1 on your touch-tone phone. Please ensure that your line is unmuted and please record your name when prompted so that I may introduce you to ask your question. Our conference is being recorded, and if you have any objections, you may disconnect at this time. I will now turn the conference over to your host, Ms. Irene Ahir. Ma'am, you may proceed.
Thank you. Hello, and welcome to today's FDA webinar. I am Irene Ahir of CDRH's Office of Communication and Education. On March 12th, FDA published the final guidance document titled Reprocessing Medical Devices in Healthcare Settings, Validation Methods and Labeling. The focus of today's webinar is to help manufacturers understand the recommendations described in this final guidance document and provide further clarification where necessary. Your presenters are Geeta Jain, a Senior Science Health Advisor from the Office of the Center Director, and Elaine Mahal, a Scientific Reviewer from the Office of Device Evaluation. Following the presentation, we will open the line for your questions related to topics in the guidance only. Please note that questions about specific products should be directed to the appropriate review office. Other center subject matter experts are also available to assist in the question and answer portion of our webinar. Now, I give you Geeta.
Thank you, Irene, and good afternoon, everyone. I'm Geeta Jain, a Senior Scientific Advisor in the Office of the Center Director at CDRH. As Irene mentioned, the purpose of this webinar is to go over the major topics in FDA's final guidance on reprocessing medical devices in healthcare settings, validation methods, and labeling. At the end of the presentation, we will also clarify manufacturers' questions on information provided in the guidance document. The draft of this guidance was published and opened for public comments in 2011. We received 487 public comments on the draft. The comments came from various stakeholders, including device manufacturers, test labs, trade and professional organizations, and users. A common theme in the public comments was the request to provide additional information and clarification on various concepts and sections in the document. For example, there were requests to better clarify the scope of the document, the 6 criteria for reprocessing instructions, and few other sections. There were also comments requesting that FDA provide more information on resources and references for developing reprocessing instructions. All comments were carefully considered as this guidance document was finalized and published last week. This is an outline of today's presentation. So, the focus is the final guidance document titled Reprocessing Medical Devices in Healthcare Settings, Validation Methods and Labeling. First, we will take a look at the purpose of the guidance document and the scope of devices it covers. Then, we will go over the major topic areas covered in the document. This includes an overview of reprocessing, FDA's 6 criteria for developing reprocessing instructions, and recommendations for validating the cleaning process. And finally, we will go over a summary of key messages from the guidance document before we move on to the question and answer session. So, what is the purpose of this guidance? As we know, devices that can be reused are commonplace in healthcare settings. To reduce the risk of patient infections during reuse of these devices, it is important to reprocess them properly between use. And a device manufacturer's reprocessing instructions are critical to ensure their proper reprocessing. This guidance document provides recommendations to medical device manufacturers for developing reprocessing instructions that can be easily understood as well as followed by users. This is to help ensure that reusable devices remain safe and effective for use on patients after reprocessing. This guidance document also outlines the agency's current recommendations to manufacturers on how to conduct scientifically sound testing to validate the reprocessing methods and instructions. In addition, this guidance also describes new measures the agency is taking to enhance its oversight of the reprocessing of reusable devices. For example, in Appendix E of the guidance, FDA has identified a subset of medical devices that pose a greater likelihood of microbial transmission and represent a high risk of infection if they are not adequately reprocessed. So the 510(k) submissions for these devices in Appendix E should include protocols and complete test reports of the validation of the reprocessing instructions. And finally, this final guidance document replaces FDA's 1996 guidance document titled Labeling Reusable Medical Devices for Reprocessing in Healthcare Facilities. So let us take a look at what is new in this guidance document when compared to the 1996 guidance document it replaces. So reprocessing, as you know, is a complex process that starts with cleaning followed by sterilization or disinfection. And the entire process needs to be validated to ensure it works properly. Recommendations in this 2015 guidance document have been expanded to include information applicable to validation of reprocessing methods and instructions. For example, specific emphasis is given to the importance of proper cleaning to ensure that the device is adequately prepared for the subsequent sterilization or disinfection steps. Recommendations are also provided for proper validation of cleaning methods as well as instructions for cleaning. This document also emphasizes the importance of device designs that are less challenging to reprocess. And it also provides recommendations on human factors considerations that are important when validating reprocessing methods and instructions. Another new aspect in this guidance is that it provides greater clarity on what documentation needs to be provided in the different types of pre-market submissions that fall under a 510(k), a PMA, a de novo, an IDE, or an HDE. So next let us look at the device type for which the recommendations in this guidance document are applicable. The recommendations in this guidance are applicable to 4 types of devices. First, reusable devices that are supplied as sterile to the user and requires the user to reprocess after each patient use. Second, reusable devices that are supplied as non-sterile to the user and requires the user to process prior to the initial use of the device as well as reprocess after each use. Third, reusable devices that are intended for use by a single patient and are to be reprocessed between each use. And fourth, single-use devices that are supplied as non-sterile to the user and requires the user to process prior to its use. Now I turn it over to Dr. Elaine Mayhall, who will walk you through the major topic areas covered in the guidance, starting with an overview of the various steps involved in reprocessing.
Thank you, Geeta. Good afternoon. This is Elaine Mayhall. I'm a reviewer in the Infection Control Devices branch in ODE. This diagram is an overview of reprocessing from the point of use processing step to cleaning and finally to disinfection or sterilization. Cleaning, disinfection, and sterilization are distinctly different processes and should be validated separately and independently. Point-of-use processing is the initial cleaning steps that are intended to prevent drying of soil on the device surfaces prior to cleaning. Cleaning is the physical removal of soil, and disinfection and sterilization are intended to kill microorganisms. In recent number of years, there has been an evolution towards the development of more complex devices with designs that are more difficult to reprocess. However, there has also been significant advances in knowledge and technology involved in reprocessing reusable medical devices. The recommendations in this guidance reflect the scientific advances in these areas. The formulation of instructions for reprocessing a device start in the early stages of device design and engineering. For example, device designs that pose a significant challenge to reprocessing include shaft within lumen configurations, fine channels, seals, and mated articulating surfaces. Less challenging alternative designs include single-use parts, flush ports, and dedicated cleaning accessories. Manufacturers should consider using designs that facilitate cleaning, disinfection, and sterilization methods that are easily and effectively implemented by users. Device labeling should provide instruction on how to prepare the device for the next patient use and should identify materials and equipment that will be needed and are readily available to users. Also, in developing your reprocessing instructions, you should consider human factors, including the use of consistent reprocessing instructions for all devices of a type. Using consistent terminology and the same document layout will improve the user comprehension and adherence. You should address any known post-market human factors issues that exist for reprocessing your device or similar devices. For example, actions requiring substantial dexterity, strength, good visual acuity, or familiarity with uncommon practices present human factors challenges. As part of your design controls, you should validate your reprocessing instructions to ensure that users will thoroughly understand and follow them. The guidance document includes recommendations designing a human factors study of reprocessing instructions. As in the 1996 guidance document, this new guidance document includes the criteria for clear reprocessing instructions that will ensure users understand and correctly follow the reprocessing instructions. The list of criteria have been condensed from 7 criteria to 6 criteria. The reprocessing instructions should reflect the intended use of the device, should advise users to thoroughly clean the device, should indicate the appropriate microbicidal process for the device, should be technically feasible, and include only devices and accessories that are legally marketed. Finally, the instructions should be comprehensive and should be understandable. Criterion 1, the reprocessing instructions should reflect the intended use of the device. The appropriate reprocessing instructions depend on the physical design of the device, the intended use of the device, and whether it has direct or indirect contact with the patient. They also depend on the type and extent of soiling and contamination the device is exposed to during clinical use. In addition, It depends on the use of disinfectants or other chemicals that might leave harmful residues or adversely affect device materials or performance if inadequately rinsed, and any specific or unique risks to the patient or the user. Criterion 2, Reprocessing instructions for reusable devices should advise users to thoroughly clean the device. Cleaning is the first step in reprocessing. Adequate sterilization or disinfection depends on the thoroughness of cleaning. If it cannot be cleaned, it cannot be disinfected or sterilized. Details of the cleaning procedure will vary depending on the complexity of the device and may include disassembly of the device, use of protective covers to reduce soiling, and flushing of the device internal structures. Criterion 3, pre-processing instructions should indicate the appropriate microbicidal process for the device. Your instructions should be consistent with current infection control principles. The microbicidal process recommended is dependent upon the intended use of the device and is described by the Spalding classification for critical, semi-critical, and non-critical medical devices. Critical devices are introduced directly into the bloodstream or contact a normally sterile tissue or body space during use. Examples of critical devices include surgical instruments, irrigation systems for sterile instruments in sterile tissues, endoscopes used in sterile body cavities such as laparoscopes, arthroscopes, and intravascular endoscopes, and all endoscope biopsy accessories. Microbial transmission from critical devices and the risk of infection if the device is not sterile are likely. These devices should be disassembled if possible and thoroughly cleaned and sterilized after each use. Semi-critical devices contact intact mucous membranes or non-intact skin, but do not ordinarily penetrate tissues or otherwise enter normally sterile areas of the body. Examples of semi-critical devices include endotracheal tubes, bronchoscopes, laryngoscope blades, and other respiratory equipment, esophageal manometry probes, diaphragm fitting rings, and gastrointestinal endoscopes such as duodenoscopes. These devices should be thoroughly cleaned and then sterilized. Intact mucosal surfaces are relatively resistant to small numbers of spores. Therefore, if sterilization is not feasible, then semi-critical devices should be high-level disinfected following thorough cleaning. Non-critical devices contact only intact skin and do not penetrate it. Examples of non-critical devices include blood pressure cuffs, stethoscopes, and skin electrodes. Also, devices that do not directly contact the patient but may become contaminated with microorganisms and organic soil, including blood or body fluids, during patient care, are considered to be non-critical devices. Examples of these types of devices include infusion pumps and ventilators. These devices may not be visibly contaminated. However, they should be thoroughly cleaned and then receive intermediate or low-level disinfection, depending on the nature and extent of the contamination. Non-critical devices that may be contaminated with blood or body fluids that may contain blood-borne pathogens should be cleaned and then receive intermediate-level disinfection with a product that has an EPA-registered claim for activity against hepatitis B. Criterion 4, reprocessing instructions should be technically feasible and include only devices and accessories that are legally marketed. Reprocessing instructions should be technically feasible in the intended location, such as in the home or in the healthcare setting. The equipment and accessories needed to implement the instructions should be available for users to obtain. For example, the type and size of the brush required to clean a device lumen should be readily available. Also, the type of sterilizer with manufacturer-validated sterilization cycle parameters and accessories should be available to the users. For example, sterilization instructions should not recommend radiation sterilization for reusable devices because radiation sterilization generally is only used in manufacturing facilities. Steam sterilization is the most common method of sterilization used in healthcare facilities. Ethylene oxide, hydrogen peroxide, ozone, and liquid chemical sterilization processes are also available in some healthcare facilities. Sterilization methods and parameters should be technically feasible. Extended cycles is an example of cycles that are not technically feasible. The specifications for these cycles deviate from those found on commonly used FDA-cleared sterilizers and have limited or no FDA-cleared sterilization accessories. They typically include longer exposure times and/or higher or intermediate temperatures. These limitations can pose serious technical challenges in healthcare facilities. Criterion 5, reprocessing instructions should be comprehensive. Comprehensive instructions enable the user to understand precisely how to implement the entire reprocessing procedure safely and effectively. The instructions should include all of the following elements: special accessories and any special protection during reprocessing, such as valves, plugs, or stoppers to prevent ingress of harsh chemicals or water, special tools, sizes and types of brushes, flush port connectors, and connector size specifications. Point of use processing or pre-cleaning instructions should be included. The instructions should include disassembly and reassembly instructions, including step-by-step instructions with visual aids, and should indicate whether the device should be reassembled before or after sterilization. The method of cleaning. The instructions should include a detailed validated method for cleaning. The information should provide a list of parameters, including duration of each processing step, temperatures, and water quality. You should list the cleaning agent or the class of cleaning agent used in your validation testing. The labeling should include information on its preparation and use or refer the user to the manufacturer's instructions. You should include instructions for rinsing the device following cleaning to remove processing chemical residues. The instructions should identify the type and quality of water that should be used and the duration, volume, and temperature of the water. You should identify lubricating agents if required. Validation of reprocessing methods, including disinfection and sterilization, should include the lubricating agent. After cleaning, the labeling should instruct the user to visually inspect the device for cleanliness, and if the device is not visually clean, the instructions should advise the user to repeat the cleaning process or to dispose of the device. The labeling should include instructions for drying the device after processing and before storage. You should identify the method of disinfection or sterilization, including the validated cycle parameters and the accessories that should be used. You should provide information on specifications of the device designed to be sterilized and the packaging and load characteristics. Labeling should include instructions for reducing sterilant residuals following sterilization by ethylene oxide, hydrogen peroxide, or other processes that may leave sterilant residuals on the device. For example, rinsing is used for liquid chemical sterilants and aeration for ethylene oxide. Instructions should include drying the device. Devices should be thoroughly dried after processing and before storage. The labeling should either inform the user how many times the device can be reused based on testing, or provide the user with a mechanism or method to ascertain whether the device has exceeded its use life, including a method to establish that the device is still within performance specifications. Label instructions should include additional recommendations, such as for devices that are initially supplied non-sterile to the user and require the user to sterilize the device before use. These devices should be labeled non-sterile directly on the individual device packaging to ensure non-sterile product is sterilized before use. For a device intended to be maintained by a patient or lay care provider, the labeling should include reprocessing instructions that are understandable to a layperson and can be performed at home. Labeling may also refer the user to guidelines from professional organizations or OSHA. And of course, the instructions should include the manufacturer's contact information. Criterion 6, reprocessing instructions should be understandable. The instructions should be clear and legible. The instructions should be presented in a logical sequential order from the initial processing step through the terminal processing step, and should be described using as simple language as possible. The instructions should be sufficiently detailed to explain the correct procedures for all steps. The use of charts, diagrams, and pictures that can be posted in the workstations are helpful in ensuring adherence to reprocessing instructions. Now we turn to reprocessing validation and the cleaning process. You should conduct validation activities to demonstrate that your methods are adequate to allow the device to undergo further processing to eventually be reused safely, and that your reprocessing instructions are effective in conveying the proper reprocessing methods to the user. This section is significantly different from the 1996 guidance document and provides information on the methodology recommended for cleaning validation studies. Your validation testing should include the worst-case implementation of the cleaning process. Use soils that are relevant to the clinical use conditions of the device and should include predetermined cleaning test endpoints. The artificial test soil should represent materials that the device would likely be exposed to during actual clinical use and would create the greatest challenge to cleaning. For example, during visualization of the larynx, a laryngoscope would likely be exposed to both blood and mucus. The artificial test soil should be a multi-component soil that includes substances that simulate both blood and mucus. The application of the test soil should mimic worst-case clinical conditions, and the device should be inoculated in all locations likely to contact patient materials, especially all locations that are difficult to clean, such as mated surfaces, lumens, and hinges. The test devices should be exposed to simulated use conditions that incorporate multiple full use cycles, assess the accumulation of soil over time, and account for real-world use conditions to mimic worst-case clinical use. For example, you should conduct all functional procedures, such as repeated articulations, flexures, and manipulations, in order to soil the device sufficiently. Powered handpieces and electrosurgical instruments should be powered on to simulate use conditions. The validation protocols should use, for example, the shortest times, lowest temperatures, and weakest solutions for each step of the cleaning instructions. For example, if the instructions recommend a 10 to 20-minute pre-soak, the validation protocol would specify 10 minutes as the soak time. A side-by-side comparison of the labeled cleaning instructions and the cleaning process used in the validation protocol should be prepared and provided. FDA recommends that you use at least 2 quantitative test methods capable of directly measuring clinically meaningful levels of the test soil to meet a relevant, predetermined cleaning endpoint. For example, protein, total organic carbon, and hemoglobin assays. You should validate the quantitative test methods you choose to measure residual soil, including for analytical sensitivity and specificity. Exhaustive extraction and extraction using a known quantity of soil are commonly used extraction methods. Recovery efficiency should be determined as part of the validation of the extraction method. All surfaces should be sampled, including internal surfaces and mated surfaces. You should use an appropriate extraction volume to remove the test soil from the device. For example, if the volume is too large, the test marker will be diluted below the level of detection for the assay. Testing should include appropriate controls, including a negative device control, which is unsoiled but undergoes the same cleaning and extraction process as the test device, a positive device control, which is soiled with a known amount of soil but is not cleaned and residual soil is extracted, a negative sample control, which is an extraction with no device and serves as a blank, and a positive sample control, which is an extraction with a known amount of soil but with no device. This last control serves to address interference of the extraction fluid and extraction methods with the soil detection. The microbicidal processes should be validated. For example, for disinfection, you should demonstrate that the device can be disinfected to the appropriate disinfection endpoint using contact conditions that are consistent with those specified in labeling for the legally marketed disinfectant and under worst-case conditions. For sterilization, FDA recommends validation of cycle specifications that are consistent with the conventional parameters as listed in Appendix C of the guidance document. For pre-market review, FDA will review the reprocessing instructions included in the labeling when we review pre-market submissions for reusable medical devices. All cleaning, disinfection, and sterilization procedures should be validated, and validations should be completed prior to submission of your pre-market application. For PMAs, HDEs, and de novos, protocols and complete test reports of validation of reprocessing instructions should be submitted. For 510Ks, FDA expects manufacturers of a subset of devices listed in Appendix E to include data in 510K submissions to validate their reprocessing instructions. Validation data may be also requested as needed for substantial equivalence for other devices. For IDEs, a summary of the validation reprocessing instructions and methodology should be provided. The key messages of the guidance document are that manufacturers should provide adequate labeling that includes instructions for reprocessing and reusing devices and device accessories safely. All cleaning, disinfection, and sterilization procedures provided in the labeling should be validated. Finally, the labeling should provide sufficient instructions on how to prepare the device for the next patient use. Manufacturers should identify the materials and equipment that the users will need to reprocess the devices, and these materials and equipment should be readily available to users. That concludes my formal presentation, and now we will take questions from the audience.
Thank you. At this time, if you wish to ask a question, please press *1 on your touch-tone phone. Once again, if you would like to ask a question, please press *1. Please ensure that your line is unmuted, and please record your name to be introduced for your question. Please stand by for our first question. Our first question is from Pentos Medical. Your line is open.
Great, thank you. We really appreciate FDA's guidance regarding reprocessing. This is obviously a challenge for a lot of companies, and we appreciate the opportunity to talk directly to you about the guidance document. One thing that FDA noted in the guidance was that all relevant internal surface areas should be sampled during the extraction method, and in some cases, additional disassembly processes may be required to adequately extract residual soil. So what we're wondering is if FDA could describe its expectations specifically regarding side viewing duodenoscopes, and whether or not the expectation is different for these scopes compared to other endoscopes.
Thank you for your question. So the expectation is applicable to all reusable devices. It's not applicable to any specific category. The recommendations in this guidance are applicable across the board to all reusable devices in general.
Okay, great. And with regard to the requirement for the potential for disassembly of the devices during cleaning, can you speak a little bit about that and when that might be required?
This is Elaine Mayhall. I think in general we wouldn't expect you to do disassembly beyond the normal disassembly that the user would be doing for the device. So we wouldn't expect destructive testing unless you could, there may be a situation where you have to demonstrate that liquids are not getting through or past those mated surfaces and into other areas. But in general, we wouldn't expect you to do destructive testing.
So in the example you just provided, are you talking about liquid seeping beyond, for example, O-rings?
Right.
So in that case, would you expect that we disassemble the scope to the point of the O-rings? Because that's not something that the user would typically do.
I think there's some special situations, and I think that's really beyond the scope of this discussion right now. So you can always address that to the review office for the device.
Okay, thank you.
You're welcome.
Operator, we'll take the next question.
Next question is from Sanjay Parikh. Your line is open.
Yeah, hi. Thanks for the good presentation. In Section 5, it specifies that manufacturer should provide the contact information. Now, in the contact information has to be domestic, as our parent company is in Denmark, Copenhagen, and the instruction for use has their address. So it is mandatory that we should use our U.S. address in the IFU or the instruction for use.
Could you please repeat the question?
Yes, sure. In Section 5, it specifies that manufacturer should provide their contact information. The contact information has to be domestic or has to be U.S. The contact information has to be the U.S. address or the U.S. phone number?
Correct. It should be U.S. The contact information for the manufacturer?
Yes.
Okay. I don't think it has to be a U.S. Contact, as long as the user can contact the company overseas. That would be fine.
Okay. Thank you.
Our next question is from Terry McMahon. Your line is open.
Hello there. I work with a contact lens manufacturer, and I've got actually two questions to ask. And the first one would be a contact lens, a gas-permeable contact lens, would have some exposure to mucus, but have you actually categorized it as semi-critical or the non-critical? Do you know?
Thank you for your question. However, it appears to be a device-specific question, so we recommend that you submit your question to DICE. The contact information is provided on the slide that is up there.
Okay. Tell me again who I'm supposed to contact. I'm sorry.
It is the Division of Industry and Consumer Education, and the email is D as in David, I, C as in cat, E as in Edward, at fda.hhs.gov.
Very good. Thank you for that. It's most helpful. And the second question is you said that, that you had to indicate that the products that you were using to clean and disinfect the product, it should say that it's a legally approved product. Now, does that actual term need to be on the instructions for use, or can you shorten it by saying like FDA approved, or does that term itself need to be on there?
Are you talking about a disinfectant used on the contact?
You said during the webinar that you had to indicate that the product that you're using to clean, sterilize, disinfect needed to be legally approved. And I'm wondering if that's an exact term that needs to be on the instructions for use.
Now, the disinfectants are, the high-level disinfectants, liquid chemical sterilants are FDA cleared. The low-level and intermediate-level disinfectants are Class 1 devices and need to be legally marketed per FDA, but they're also regulated by EPA, so they should be registered by EPA. So you don't have to say in the labeling that these to specify that the product is legally marketed. We'll look at that when we look at the review, but you should specify a type of disinfectant that is legally marketed.
Okay, so as long as we specify a brand or something along that line and it is approved, then we're good.
Correct.
Okay, very good. Thank you.
Our next question is from Diane King. Your line is open.
Hi, thank you very much. I have three questions. The first is from Criterion 5, the section that refers to reduction of sterilant residuals. it refers specifically to EO and to hydrogen peroxide, and there's a reference to ISO 10993-7 for acceptable levels of EO residuals. Do you consider that acceptable levels of hydrogen peroxide residuals have been established, and if so, do you have a publication to which you refer to those?
Hi, this is Steve Elliott. Currently, there are no established levels for hydrogen peroxide residuals. Concerns associated with hydrogen peroxide should be addressed through material compatibility of the device and biocompatibility. So that would largely be device specific to provide that information. To my knowledge, that specific ISO standard doesn't contain any information along those lines either.
Okay, great. My next question involves whether the validations or the test results that the agency would expect to see in a 510K would cover the possible effects of combining or alternating between different validated sterilization methods over the life of the device.
That is a complex question. I think that's something that might be needed to be addressed in more detail with the device-specific office, possibly through the pre-submission process.
Okay. All right, great. Thank you. My last question pertains to Criterion 2, which mentions the use of protective covers and states that cleaning instructions should assume the worst-case situation in which the device is used uncovered. I'm wondering whether the agency considers a scenario in which performance criteria for a device-specific protective cover could justify use of a critical device after disinfection rather than sterilization.
I think that's more specific to the device. So it should be, you should talk to the device-specific office. But in general, if a cover is used, whether it's it's considered to reduce the soiling and to make it easier to clean. So if it's supposed to be sterilized, it should still be sterilized. There are a few exceptions to that, but there's specific devices with specific performance testing and so forth. So it would be best to talk to the device-specific office on that.
Okay, great. Thank you.
An additional comment with regard to the ethylene oxide residuals. The guidance document does refer to AMI ST41 and, as you mentioned, ISO 10993-7. The most helpful information you'll find will probably be in AMI ST41 for designing an aeration procedure.
Okay, thank you.
Thank you. Our next question is from Porte Tola. Your line is open.
Yes, hi. Thank you for the very good presentation. I have three questions for you. One is about the different cleaning agent. If you are recommending several cleaning agents, do you have to validate using each individual one or just the least effective?
Could you repeat the question once more, please? We were not able to hear you.
Oh, sorry about that. No, it's about if you are recommending several cleaning agents, do you have to validate each individual one, or is it enough to validate the one considered the least effective?
Okay, cleaning agents are a little bit difficult because there's no standards for them and no regulation of them. So in general, if you validate with a specific cleaning agent, you should reference that cleaning agent in your labeling or the similar class of cleaning agents, such as an enzymatic cleaner if you used that. I don't know that you have to validate with every cleaning agent, but if you mention it in your labeling, you probably should do validation testing with each agent.
Oh, okay, good. That's understandable.
And one other thing in addition to that, if you have questions about which one's going to be most or least effective, any validation design plan should really be put together such that you're testing worst case. So if you have an option in there, for example, the detergent says, the cleaning agent says mix, for example, 1 ounce per gallon, you want to design your validation protocol so it's testing worst case. So you want to do 1 ounce per gallon at the most, but really to prove what's going on there, you'll be better off going a little below that.
Oh, okay, good. Yes, then the next one is about the number of cleaning cycle. Do you have any recommendation about how to select the number of cleaning cycle you want to use for your validation?
We don't provide any specific, recommendations on specific number of cycles.
Okay.
I mean, if this question is related to any specific device and in the scenario in which that is applicable, we recommend that you touch base with the appropriate review division.
Okay. And I think it might be the same thing. How many sample will be most appropriate? We are talking here about non-critical devices.
Again, that would depend on what kind of device you're talking about. So, that would be a question that you should reach out to the review division.
Okay. Okay, good. That's fair enough. Okay, thank you very much. And that was very important information.
Thank you. Our next question is from Robert Thomas. Your line is open.
Thank you. Yeah, this is about the cleaning procedure. I'm actually a chemist who has worked in the field of chemical detergency for many years, and I think it's well accepted that the cleaning chemistry for ultrasonic type cleaners have not been optimized for cleaning medical devices. They're typically generic enzymatic type cleaners that were designed for other cleaning applications. And I was wondering if the FDA are going to make recommendations on specific cleaning chemistries that are better suited for removing biological debris.
We recommend that you select and validate whichever cleaning method is applicable to your device and provide that information in the device labeling.
Okay. The manufacturers of the ultrasonic cleaning equipment, handle that, and the chemical supplier doesn't often have a say in what type of cleaner is used. That's my only concern. And it's it's clear to the organization that I represent that the cleaning chemistries could be optimized, could be improved, and it's a I think it's kind of well accepted in the field that you could reduce and improve the cleaning cycle with different chemistries. So that's the point I was making. I just wondered if the FDA would be making recommendations based on the cleaning chemistry, or is that something that you wouldn't do?
Okay, so that is a specific question related to certain cleaning chemistries and technologies, and we recommend that you submit your questions to DICE, the Division of Industry and Consumer Education. Again, the contact information is on the slide that you see up there.
Oh, okay. All right.
Thank you.
Okay, thank you.
And one additional comment on that. It's different cleaning chemistries are going to be more effective or less effective depending upon what they're supposed to be cleaning. So it depends on, as you'll see in the validation sections, there's emphasis on picking clinically relevant soils to simulate contamination and use. And the cleaning chemistries that should be chosen should actually address what those contaminants are. So if something is going to be very high in protein or if something's going to be very high in carbohydrate, and you're choosing a different type of enzymatic cleaner, you want to choose the types that are lipases or proteases or whatever it is that you're looking for. So,
yeah, and even perhaps if an enzymatic cleaner is not appropriate, that there are many cleaners based on ionic strength that work better, without enzymes being present. So that's the whole point I'm making, that there are lots of different chemistries out there, and I think probably they haven't been optimized for the removal of sort of biological debris. I mean, as a chemist, you look at these sort of, cleaning capabilities, and you know that the cleaning process could be improved.
Right. The cleaning process is important, and there's a lot of different methodologies that can be used, but it's really much more detailed than what we discuss in the guidance document, and it needs to be addressed in validation testing for the individual devices.
Okay. All right. That's good to know. Thank you.
Thank you. Our next question comes from Michelle Alpha. Your line is open.
Thank you, and just wanted to commend the group. It was a great presentation and helped clarify a lot of things. You mentioned under the cleaning validation section that worst case conditions should be mimicked during the validation process, and you also indicated the use of 2 quantitative test methods. And I was wondering if you could comment on devices such as flexible endoscopes that go into the colon and get exposed to microbes in addition to organic material. If the enzymatic or the detergent that is being used in cleaning doesn't kill the vegetative bacteria, could your markers be, for instance, a protein quantitative marker and then a microbial viable level marker? I do appreciate that the guideline states very clearly spores aren't to be used, and that makes total sense. But since the purpose of the cleaning is also to reduce the level of microorganisms present, I wondered if one of the markers could be viable microbes.
Pretty much we recommend against using any kind of microbial log reduction for demonstrating cleaning because we haven't seen adequate data that correlates the reduction of microbes on the device to removal of soil. So in general, we're not going to, unless someone can provide data demonstrating that there is a correlation, we don't expect to see the use of microbial log reduction for cleaning validation.
Okay. The only reason I asked the question is the objective is really to make sure the microbe load is low enough so that when you do disinfection, it will be expected to be effective. But I understand the comment that you made. And the last question I had was, I noticed that when you were talking about the Spalding classification, you have categorized bronchoscopes as semi-critical. Could you expand on that a little bit, given that they do enter a sterile body site in terms of going into the lungs?
Well, in order to get to the lung, they have to go through the larynx and the throat. So you really should, and it contacts mucosa. So it's really a semi-critical device.
I understand that, but I guess it's a bit confusing when, for instance, biopsy forceps that would go through an endoscope and may also go over the mucosal surface, the expectation is it should be a sterile device. But appreciate your comments back on why it ended up in the semi-critical category.
Right. Well, the use of the biopsy forceps may be a different situation in that case. But that would be a question really for the device review division.
Thank you.
Our next question is from Anita Krzyzewska. Your line is open.
Thank you for your thorough presentation, FDA. We have two questions from Tanovis. First one is, does FDA have a position and preference for representation soil for fidelity to superbug cleaning sterility of validation?
Could you repeat your question one more time, please?
Certainly. Does FDA have a position and preference for representative soil for fidelity to superbug cleaning sterilization validation?
Okay, so as you can see in the guidance document, we do not, we do not provide recommendations on what specific soil to select. We ask that, whichever soil you select, it needs to mimic the conditions of use of the device or the clinical use conditions of the device. Thank you.
Okay. Second part of the question is for positive device controls, does the new guidance address the soil accumulation over time and or provide suggestions for methods on device-specific collection and simulation?
Hi, this is Steve Elliott. With soil accumulation, this is something that is expected to be addressed through validation testing. However, it is something that might vary from device to device, so it could be a device-specific concern related to issues cleaning and reprocessing a specific device line or device type within that. So probably that is a question that would need to be addressed by the device-specific office.
Okay, thank you. And lastly, I had a question surrounding also the post-human factors labeling. Can you expand a little bit more detail on that information?
Could you be a bit more specific on, we have a couple of paragraphs on the human factors, so what specifically are you looking for?
So what type of, when you say post-human factors, that to me is implying the post-commitment marketing requirements that we have to monitor. Is that something that we have to execute after we get the device cleared?
So basically what we are saying is, if you are aware of any issues, any post-market issues that have occurred, those considerations need to be taken into account.
Okay.
And, if you look at page 7 of the guidance, we have provided a couple of examples where, you should address the specific aspects such as, actions that require substantial dexterity or strength, good visual acuity, familiarity with uncommon processes. So those are the kind of things that need specific attention when you validate and provide your reprocessing instructions.
Okay. Thank you. Thank you so much.
Our next question is from Shannon Scruggs. Your line is open.
Thank you. Good morning, or afternoon, I guess, at your time. I just wanted to be sure because you may have said it at the beginning of the presentation, but we wanted to be really clear. Is the new guidance applicable only to new reprocessing devices, or is it applicable to all of our approved devices, even those approved before 1996?
Hi, this is Sergio Del Castillo from the Office of Device Evaluation. The guidance will now be in effect for any submission that comes into house as of the March 12th date of the guidance.
So any new submission as of the 12th? Okay.
Correct.
All right. Thank you. That was the only question that I had.
Our next question is from Sandra Abrahamian. Your line is open.
Hi, I have a question about single patient reuse devices that was added to the scope of the guidance document, because it was added into the scope but not really addressed within the guidance document itself, except through the process overview and the appendix. And so the question is the flow chart goes into the risk of cross-contamination of the device, and then also if the device could be soiled by body fluids. And it uses kind of vague terms, unlikely or maybe. And so I was kind of wondering how the agency is going to make those determinations for devices that are specifically labeled as single patient use.
Hi, this is Steve Turtel. It's really, again, this harps back towards the way the validation protocol should be designed, and that should be for worst case scenarios. So if there's any doubt, that there might be accidental use or just rare occasional use of there being a contamination event occurring, the design of the validation protocols really should be put together and written in such a way that they address the worst case. So if the contamination event that may occur, it should be included really in the design of the validation protocol.
Plus, that would be a good question that should go directly to the device review division.
All right, thank you.
Our next question is from Ed Marandola. Your line is open.
My question is actually very similar to the last and probably was answered by the last. But in the scope of the guidance where you talk about reusable devices intended for reuse by a single patient and reprocessed between each use, do you guys define what a use is, or is there a definition of what a use is? Is there like a time period to it? Is it
we do not define the word use in the guidance, but a use is the use of the device. And maybe, maybe for certain specific devices, if you have questions, again, you please refer the question to the specific device review division.
Okay. Thank you.
Our next question is from Julia Ye. Your line is open.
Hi. I have a question regarding the non-critical devices. It gives examples, like a stethoscope or blood pressure cuff. I wanted to understand what type of validation would be required for those types of devices, because they don't actually, or my understanding is they wouldn't actually involve, determining the microbes and soil or validation of those types for this non-critical device. Can you expand on that?
The labeling should include instructions for reprocessing any type of reusable device. So even for a blood pressure cuff, there should be instructions for cleaning and disinfection of the device, because it could become soiled with patient material.
Okay, so there would be an expectation to validate that cleaning. Let's say if it was with like an alcohol swab, there would be an expectation to validate that the alcohol swab removed soil from this type of device.
Correct.
Okay, thank you.
Our next question is from Sarah Friedberg. Your line is open.
Hi, thank you for taking our questions and for the presentation. I had a question on Criteria 6, that the instructions should be understandable. One of the recommendations is that we, as manufacturers, include charts, diagrams, or pictures that can be posted in workstations, and we completely agree that having, a diagram of some of the more complicated cleaning steps or sterilization processes could be helpful for the end user. I guess my question is, expectation-wise, for something like a chart or a diagram that's going to be removed from the IFU and placed up on a wall, what level of warnings and cautions would FDA want to be on such a chart or a diagram? Would it need all of the cautions and warnings that would be in the entire IFU, or is it sufficient to put, a statement that the IFU should be looked at to see all warnings and cautions?
I think that just depends on the specific device and what's critical to be on that poster. So again, I think that's a question that's specific to the review division for whatever device you want to ask about.
Okay, thank you.
Our next question is from Frank Canosia. Your line is open.
Good afternoon. This is Frank Canonica at Pentax Medical. I was hoping that you would be able to help clarify a description in Section 8 on the various test types that are used to validate cleaning. When the guidance states that at least 2 quantitative test methods should be used, do you mean that for each soil indicator, say protein, carbohydrate, and hemoglobin, are you saying that for each one of those types of soil indicators that 2 quantitative methods should be used to assess residual levels of those indicators? Or are you saying that only 2 indicators need be tracked, and for each of those 2 indicators that only 1 quantitative method be employed? Or are you saying something different than either of those 2 descriptions?
That would be your second description, not the very last one, but basically we want to know that of the components that are in the soil, that there's at least one test type that addresses each of those components. So for a particular clinical soil, you may have varying degrees of carbohydrates, proteins, lipids. We just want to know, we would like to see quantitative test data, for example, 1 for the protein, 1 for the carbohydrates, or if you wanted to include total organic carbon, however you decide on which those 2 are, the total of 2 is what we're looking for.
Okay, thank you for the clarification. Now, that is, from what I understand, that is a change in course from what is typically being employed at the present time. I know that historically, at least for determinations that we have conducted on cleaning, we've been following 3 markers, namely protein, carbohydrate, and hemoglobin. Is the agency saying now that 2 markers are sufficient? And if so, which is what I derived from the answer that you just provided, and if only 2 markers are required where we were normally providing 3, which of the 3 markers should we be providing, and on what basis?
So we are recommending that you provide a minimum of 2. I mean, if you use 3 and provide that information, that's fine, but our recommendation is to provide a minimum of 2, like protein and hemoglobin, or total organic carbon, any 2 quantitative markers.
Is there any 2 that are preferential? Is there, say, among protein, carbohydrate, and hemoglobin, is there a particular priority order that the agency has determined? Is one more important than the other? I mean, because if we're going to drop down to 2 instead of 3, we certainly don't want to pick the wrong 2. So is there some kind of priority that's been established among protein, carbohydrate, and hemoglobin as to which one of those 3 may be omitted from this point forward?
Whichever marker you pick, it needs to represent the conditions that the clinical, the device would be exposed to under clinical use. So if it is exposed to mucus, for example, you need to pick the appropriate marker that is relevant to assessing mucus. So if it is exposed to blood, then, hemoglobin is an appropriate marker. So essentially, we are asking that you pick the soils based on the clinical use of the device.
Okay, so that would depend on the use of the device, the particular formulation of the artificial test soil that we construct in order to represent the anatomical site where the device is used, and then the prevalent markers from that ATS formulation.
So there's several considerations there in the design of your protocol. And in general, we're giving guidance here, but a couple questions have come up regarding sample sizes or number of uses or types of soil used. And really, it's important to think it through as best you can to find out the answer, the best way to find the answer to get most meaningful data that you can. And if you choose to use 3 markers or a very large number of samples, it's only going to strengthen your data and give you better answers, more valuable and more meaningful answers. Another element of that is how you choose those particular markers for the particular soils. It may vary depending upon how, what component they are, what percentage of the component they are in the actual clinical soil. But you may also need to evaluate the degree of strength that they adhere to the surfaces. So some of those components that you mentioned, some of them may be much more tightly binding to the surface and may be more of a problem for cleaning. So there's not a one answer fits all with these things, and that's why we do, on a number of occasions, recommend that you contact the lead reviewing branch or division because they have a little bit better understanding of those particular device types.
Understood. Thank you very much. One additional follow-up question. As relates to simulated use of the device, again, historically, in simulating the use of the device, we have been employing, approximately 10 cycles under the guidance of the agency. And now that we're reading the newly passed guidance, newly issued guidance, the guidance makes a reference to employing a statistically significant number of cycles during simulated use. My question is, can a device manufacturer anticipate that the number of simulated use cycles that they will be required to perform will now evolve to something other than the standard 6 to 10 cycles that have been employed previously? In other words, are you going to have to do some kind of a calculation in which you anticipate for a reusable device that it may be used X amount of times over its useful life or under the warranty period or whatever parameter you choose to select, and then run a number of simulated use cycles that somehow relates to that number?
So I believe this question, the number of cycles would depend upon the complexity of the device and the specific features of the device. So this is a question that you should be asking the review division. They would be able to give you more guidance on that.
Okay, thank you very much. I appreciate it.
Our next question is from Daniel Rudnick. Your line is open.
Hi. In this presentation, you mentioned that cleaning and sterilization are treated independently. So I just wanted to clarify that there's no expectation that every combination of cleaner and disinfection and sterilization process is required to be tested.
You should be testing whatever you recommend in the labeling of the device.
So, for instance, if we recommend 6 different cleaners and 3 different sterilization processes, Do we have to test every combination of those, because that becomes an exhaustive number of tests?
Yes, you should do validation testing to validate the cleaning process with anything that you specify in the labeling, and you should definitely do validation of each of the sterilization methods recommended in your labeling.
Yes. So we test every method, but we're not required to test every combination. In other words, cleaner 1 with sterilization method 3, and then cleaner 1 with sterilization method 2. You treat them as independent.
To validate the cleaning method and the sterilization methods, they have to be validated separately and independently. If you're looking at biocompatibility or compatibility of the device with the processes, that's different.
And then again, whatever you're recommending in the labeling, that is what you need to test and validate. I mean, you asked a question, I believe, like cleaning method 1 with a certain sterilization method. So if that is what you're recommending in your labeling, that needs to be validated.
Sorry, it's not clear to me. If we're recommending 6 different cleaners and 3 different sterilization methods, and we test each one independently, that's sufficient?
Again, that depends, if you're recommending 6 independent cleaners independently and 3 disinfectants independently, and it does not matter how the user uses them, you can validate each independently. Then again, it depends on, what specific instructions are you providing in your labeling. Whatever instructions they're providing in your labeling, that needs to be validated.
Okay, thank you.
Our next question is from Charity Hobie. Your line is open.
Are there any requirements for a microbial log reduction for each disinfection level, such as low, intermediate, or high?
There are. If I can remember it right now, it's not specified in the guidance document. You may be able to find it in other guidance documents, such as the one for medical washer disinfectors. Low-level disinfection would be a 6-log reduction of 4 vegetative bacteria, Klebsiella, Pseudomonas. I can't remember the other ones, but there's 4 different ones. And then for intermediate level disinfection, it would be the 6-log reduction of each of those vegetative organisms, as well as showing a 3-log reduction of an appropriate mycobacterium species. And each of those tests should be done independently, separately with each bug. And then with high-level disinfection, you should show a 6-log kill of mycobacteria.
And so you can't do a cocktail of microbes?
It's preferable to use, to test each organism separately. If you can provide justification for doing a cocktail, you can provide that, but generally we've requested that each organism be tested separately.
Okay, thank you.
At this time, we're going to be taking our last question. And our last question is from Gary Sokola. Your line is open, sir.
Great. Thank you for taking my question. In Section 9, it discusses disinfection and sterilization, and in the second paragraph, there's a statement, validation data should be generated in FDA-cleared sterilizers and with FDA-cleared sterilization accessories, e.g. biological indicators, physical chemical sterilization process indicators, sterilization wraps. This typically is done when validating sterilizers or sterilization processes, and when validating reusable medical devices, specifically Class 2 devices, typically the biological indicators used for the validation process are not FDA cleared, not all of them. Most healthcare FDA-cleared DIs are self-contained. So when trying to validate a complex device like an endoscope or some other lumen-type device, inoculated threads or even direct inoculum is used. So this statement appears that those types of DIs cannot be used in the validation process anymore.
Hi, this is Steve Elliott. With regards to that, the intention behind that language is to ensure that the validations are going to be consistent with FDA-cleared cycles and accessories. If appropriate, it could be acceptable to use biological indicators that have not been FDA cleared, particularly if the resistance characteristics are known and appropriate, and the nature of the validation involves something like a direct inoculation or placement on a wire or something like that that wouldn't be possible with a paper BI or SCBI.
Okay, great, thank you.
I turn it back over to your host.
Thank you. This is Irene I here. We appreciate your participation and thoughtful questions. If you were unable to ask your question during our question and answer session, please send your inquiry to dice@fda.hhs.gov. Today's presentation and transcript will be available on the CDRH Learn webpage at www.fda.gov/training/CDRH Learn by Wednesday, April 1st. If you have additional questions about the final guidance document, please use the contact information provided at the end of the slide presentation. As always, we appreciate your feedback. Again, thank you for participating, and this concludes today's webinar.
This does conclude today's conference call. We thank you all for participating. You may now disconnect and have a great rest of your day.